Hello, everyone. Welcome to the sixth MultiScale Talk given by Dr. Ali Hoshiar. Ali is assistant professor at the University of Essex in the UK. He has been working on a small-scale robotics, including actuation, sensing, and control, and mainly focusing on medical applications. Before joining University of Essex, he was a research fellow at the Storm Laboratory at the University of Leeds, also in the UK, where he was focusing on the development of soft continuum magnetic robots for surgical interventions. Ali has also conducted research stays at the DIGIST ETH, Swiss South Korean Joint Microrobotic Research Center, and GNU, also in South Korea. Today, Dr. Hoshiar is going to talk about microrobotic systems for swarm delivery of the therapeutic agents. So Ali, the screen is yours.
Okay, thank you, Thoron. So I will share my screen and, okay. Yeah.
Great.
So yeah, my name is Ali Hoshiar and I'm with the University of Essex. Today I will be talking about microrobotic system for swarm delivery of the therapeutic agents. So first of all, what I want to share with you is that if you look at the common disease, the common cause of death, as you can see in here, the four major cause of death. And then you can see here the unfortunate actually increase in disease like Alzheimer, as well as actually cancer, whereas we have some sort of success in reducing the number of deaths on heart disease or stroke. And then this is actually where we can consider this macro-nano based technology, especially for the medical application as a swarm, to do the drug delivery and then to help to treat these sorts of disease. And the second graph is actually basically showing that by 2016, these, especially the Alzheimer, would be the major cause of death. So the major cause of death by 2016 will change from what we are, the challenge that we have now. And the idea for, these sorts of treatment is coming from science fiction. So my favorite sci-fi writer is like Asimov had this book, and then it was based on a movie called Fantastic Voyage. And the whole research area of micro-nanorobotics, especially medical micro-nanorobotics, is coming from this movie. So let's start with that movie. So here what you can see is a part of the movie which is showing the concept. So in this concept, what we are doing is that they are injecting a miniaturized submarine. So this submarine will go to the blood vessels, it will reach to the brain, and it will open a blood clot, which is not, they cannot operate. So this is the idea, miniaturizing something and then putting inside the body, and then you can see the radars. It's very interesting because at the time there were no monitoring devices, so there were no MRI, and they were showing this that they can localize with using this sort of radar technology at the time. But the concept is very much similar nowadays. So yeah, let's see in a closer actually look. Let's see that how we are making that Fantastic Voyage reality in micro-nanorobotic. So you can see here, there is a miniaturized submarine and it has different elements. So one element is miniaturization. So obviously we cannot miniaturize the submarine, but we can now, because of nano-micro technology, we can miniaturize devices. And then they are using nuclear energy, which might not be a good idea in terms of health and safety. So we are using a safe magnetic field to control mostly. This is the favored method. And human operator. We keep that human operator idea, but instead of actually miniaturizing humans, we are using master slave system to control these sorts of devices. And then the part for monitoring was quite correct. So we are using external monitoring devices to localize, to find the pathway to, and guide to, the desired location in the body. And then for a specific medical application, here they had these laser guns to open a blood clot on that story. And then we are also using different approaches for the same opening the blood clot or for targeted drug delivery to a different region in the body. So if I want to move from science fiction to some sort of schematics and how this works, is that as I mentioned, we are replacing the magnetic actuation system for actuation, and then we are controlling a miniaturized devices like macro nanorobots, and then we are using external monitoring system to monitor, and then we would have two different approaches. We would have feed-forward approach, and we would have a feedback approach. And for feedback approach, nowadays we have lots of quite capable, medically approved monitoring devices like MRI, MPI, and other devices. And the next, actually, I want to talk about now different element in this system. So one element is the Microrobot, and we have different actually generation and different type of this Microrobot. So we have tethered Microrobot like this. We have untethered Microrobot, which are not connected, and then you can control it using your magnetic field. And then this is the needle type Microrobot that was developed in 2020 South Korea in DG's ETH. And then we also control swarm of them. So these are swarm of nanoparticles that you can control, you can shape, and then these are the topic that this is the topic that I will describe in more detail today. So let's talk about the electromagnetic actuation generally as a tool that we use to control these sorts of devices. And we have actually two different ways to actuate them. We are using magnetic torque, and any object which is located in, magnetic object in a magnetic field, it will try to align itself with the field, so it will face some sort of torque. And if you have some sort of gradient field, then that gradient field also is applying some sort of force, which is a gradient-based force, magnetic force. So we have magnetic force and magnetic torque to control. And then based on this, we are developing different generations of electromagnetic actuation systems. So this is one of the generations that we developed in South Korea. So we were using two magnets to control, but two very, very strong magnets. And then we did in vivo test with controlling magnetic nanoparticle with this system. Now here in the University of Essex, we are developing Helmholtz-Cohen-based system. So we are controlling it in 3D and homogeneous magnetic field for magnetic torque. And obviously there are a combination of these approaches, and the most elaborately in this actually system, which is called Octomag, was developed by Brad Nelson group in ETH. And then there are different monitoring systems. So one of the things that I will briefly mention is MPI. So MPI is also a system that we have developed. It has been under development since 2005, and we use that for swarm control or cluster control of nanoparticles and real-time monitoring them. And then obviously X-ray is also a popular way to control these sorts of devices, especially tethered microrobots. So I started from science fiction, and then I showed some image of actually the conceptual image. And then here I can show you more sort of engineering image of what's happening. So we have these different parts. So we are developing that human-based parts using a teleoperation system, master-slave system. And here we have different robotic approach. We have tethered, untethered, and then we have swarm. And then we are using also already available medical monitoring devices to control them as a feedback. I will show you some of the applications and some of the technologies that we have developed. So one of them was, as I mentioned, for very important issue, which was Alzheimer. So in Alzheimer, as you know, Alzheimer is attacking a very deep region in the brain. So it's attacking hippocampus, as you can see in this image. So hippocampus is located in deep area of the brain. So getting the drug to that area is very difficult, especially because you also need to pass through BBB, blood-brain barrier. And one of the systems that we have developed was specifically for Alzheimer's drug delivery for Alzheimer treatment. And we were using the swarm of magnetic robot or magnetic particles in order to reach to that specific position in the body. And initially we were using predictive modeling. So we were using lots of modeling approach in order to control those nanoparticles and reach to deep side in the brain. And we did in vivo tests to show that we are reaching to the targeted location in the brain in the experiments. And this is the experimental setup. So you can see we are putting a mice in the region of interest, especially the brain of that mice. And then we are drug coating the nanoparticles. And then these are different actuation schemes that we were using. And then finally, we managed to go through BBB and reach to the brain, I mean, deep region on the hippocampus, which is in this area. Okay. So again, I'm not going to go through any very detailed mathematical modeling for this, but we are using magnetic torque and magnetic force as dominant inputs that we have. And then here you can see that the relationship between the force torque and the magnetic field gradient, and obviously the property of the material that you are controlling. For the specific case of nanoparticles, we consider the aggregated nanoparticles because even though these nanoparticles have coating, they aggregate, and then we were controlling them when they are aggregated. And this is actually showing that the main force that we are using is this magnetic force, and then we are controlling with that magnetic force. So this is a gradient-based system. So it is not controlling as a rotating field or as a torque, but it's controlling the nanoparticles with magnetic force. And then we considered different approaches and different schematics. So one of that schematics was using a safe zone. So in different bifurcation, if we control the position of the particles to be in the side that we want, and because we also have the fluid flow, so it will move to the direction that we want. So we are trying to move the cluster, the swarm of nanoparticles in those areas, and then we develop a controlling scheme like this. So this is one of the initial studies that we had. So here we are controlling the nanoparticles, but here the actuation actually we are using is not good. So the particles will go to the both direction. But the next one, which is not here, obviously, unfortunately, but the next one is showing, this one is showing very briefly show that we can control the direction correctly. And then this was the initial test. Here it's better, I guess. So here in the experiment, in vitro experiments, you can see that when the control is not good, we have larger particles and then they are going to both directions. Whereas when we have a very good control, the size of the particles are smaller and almost all of them are going to the same direction. So this was the first proof of concept that we had. And then we went to more detailed simulation. So we went to Console, which is a very nice platform for these sort of studies. And then we applied the same sort of things. And then we were controlling the particles. And we showed that when we are using the approach that we proposed in the simulation, we reach a very good target. So most of the particles are going to the direction that we desire. Then after extensive simulation and modeling, we went to in vivo. And in vivo, we showed that the particles are reaching to the brain. And specifically, we showed that the particles are reach to the deep region, which is hippocampus, which is the location of that issue, the Alzheimer. And so this was the result. And then we also find out that what was the optimum condition for in vivo tests. So all the things that I showed is mostly predictive modeling and feed forward. So there is no feedback. But obviously we are roboticists, so Microrobotics is also part of robotics. So we want to have feedback. And there are many different approaches for monitoring nowadays, and then many of them are used and people are trying to use that in vivo especially. One of them is magnetic particle imaging. It's a very promising approach because everything is magnetic. And we have developed an MPI-based navigation system. So MPI, the concept is something like this. So the concept is you are exposing the particle to some sort of excitation. And that magnetic excitation is, the particle in response to that magnetic excitation will generate some sort of signal. And we are actually saturating the region, the entire region, but only in the middle of that region, in this blue area, the field is free. And if the particle is located on that area, then you can see the particle. You can actually receive that signal. You can find out that particle is in that location. So what we do is that we very fast move this field-free point into that region of interest. So it's dynamic. It is moving to, inside the region of interest, and then it's a kind of a scanner, but a very, very fast scanner compared to MRI or other techniques. And then everything is magnetic, so you don't need to have two separate systems to control and then to monitor. So it's an integrated system. So it's a very interesting idea. And then this is showing the experimental result that we published. So here is the particle that we are controlling with the magnetic field. And then here it is showing the movement and the position of the particle. And here it's in 2D. In this time, actually, we are manually moving the particles. And then at the same time, we are monitoring the position of the particles. So yeah, this is the second version. So we also talked about the feedback. And as I mentioned, we have different strategies. So this is more details, of that strategies that we are developing. So we divided different actually approaches, schematics. And what we are doing, as I mentioned, is that we are considering these safe zones, and then we are trying to move the SWAR between these safe zones. So I will very fast actually move from this work, but here we are showing the different parameters like increasing in the velocity and the effect of velocity on the number of particles. So we are actually putting some challenge and then seeing that how good the system or approach that we are proposing works and what are the limitations on that study. So this is showing that when the blood flow is very, very fast, considering the geometry of the vessel that we have used in here, we have this limitation. And then after actually a specific velocity, we would not be able to completely control the particles. So let's go to this one. So here we also continue the research, and this was one of my students' work in the University of Essex. So she developed a very nice, this is based on Alison Nakamura's open source HapKit. So she developed this HapKit device, 3D printed that, and then she was using that for controlling the nanoparticles inside a multi-branch vessel using some of those schematics that I mentioned. And then what we are now actually mostly interested is a rotating magnetic field instead of using the gradient field. Because the gradient field, as I showed initially, you need a very strong magnet. And then you need all those sorts of very heavy, bulky magnets, which are very expensive and very, very hard to develop. So we were interested in using rotating magnetic field, and then we started a direction for this. So we developed a predictive modeling platform for rotating magnetic field and the different swarm pattern for that. So the goal here is that capture all the particles and then move it as a swarm toward those directions that you want as a swarm of the movement. So here we develop a platform, both for simulations and for testing. And then we show the good relevant between our experimental result and the simulations. And here in the simulations, we are showing different patterns. So we have actually considered the different patterns. Professor Li Zhang's group in Chinese University of Hong Kong, they have developed lots of very nice experimental results. We use some of those ideas, and then we verified both with the platform that we developed and the platform that they had, the results that they already published. So it was a very nice, actually, platform which was predicting different cluster forms. So yeah, that was the design of this study. And now if I want to conclude, there are different approaches for swarm control, especially for medical application. One of most popular one is feed-forward approach. So you predict with modeling, with extensive modeling, and then you test that modeling in vitro, and then you go to in vivo to find out that what was the optimized result, what was the best result that you have in vivo. Then another very interesting topic in this area of swarm robotic for smaller scale applications and for therapeutic application at a smaller scale is developing dedicated monitoring system. So we have very nice things like MRI or X-ray, and now more and more of these tools are coming out. One of them is this MPI, and then here the nice thing and the most important thing is that it should be real time. And then again, probably the way that we are exploring this is toward more rotating based magnetic field. So we are combining both two approaches for predictive modeling and then monitoring into using it for this sort of application for rotating magnetic field. Okay. Yeah, and then obviously the work that I showed here was in collaboration and the work that we have done in the University of Essex, also with the GIST in South Korea and this is ETH, Macro Nano Robotics Center in South Korea. So yeah, thank you for giving me the time to present it. Thank you.
Thank you too. Very nice work. Very beautiful results. So I'll actually start with something very kind of abstract question. How far we are from, I don't know, having blood dissolving clot in the human body?
Okay.
Probably everyone asks you this question.
Yeah, exactly. This is a question that I asked in 2016. I asked it from Sylvia Martel. So the answer that, Professor Martel gave me at the time was 10 years. So he told me that 10 years from now, we might see something in clinical use. And 5 years passed. I can tell you that the answer is very much similar. So 10 years from now, we might see some of these in real application. So the good news is there are very, very good group and it's attracting, the topic is very hot. So we are attracting lots of research fund in this area. So that's a good news. And I can tell you that, for example, I know Pietro Baldestri at the University of Leeds, and his group, the Storm Lab group, they started doing the clinical trial for soft robots to do the colonoscopy. And this is a very good news for everyone in the medical robotic field, because it seems that, this soft robotic and also obviously a small scale robotic field moving towards preclinical and the clinical trials, which is a very good news.
Yeah, absolutely. Okay, now I have a bit more technical questions. So in this work where you had the spinning magnet, so because it's completely, it's of course certainly rotating magnetic field, but also it's not only field, you have also huge gradient if you have magnets there. Am I correct?
Yes.
So because if you have a Helmholtz coil, the point is that you have a very uniform magnetic field in the center between the two, what's the name of this, between the two coils.
Yeah.
And then if you have, like 4, then mainly the region, the RMS value is very small, how much it varies the field. So we all assume it's uniform magnetic field. But then how is the uniformity of the magnetic field when you have a rotating magnet?
I can say obviously it's not Helmholtz coil, but the unity wasn't that bad. We did the experiments. I'm not sure about the exact number, but I believe that it was about 5%, especially on that, the very much the center of the region of interest. Because when you're actually in the distance from that center, it drops. But yeah, in the center, it was probably 5% deviation in the magnetic field. So you have a gradient anyway with the permanent magnets.
Yeah. Okay. And this MPI, this the navigation, the monitoring. Are there some I don't know, any success in vivo trials, because you showed some mouse? Can you really see in the mouse where it's the magnetic particle?
Yes, not the result that we have published. Our publication was in vitro, but there are some publication for in vivo result with this MPI system. So the MPI, the idea was first introduced in 2005. So it's relatively very new. It comes from a research group, especially on physics side. And so they started that in Germany. I don't remember exactly which university, but they started it there. And now it is even actually, it's very expensive system, but it's commercially available. They commercialized a version of that, I guess, 3-4 years ago. So yeah, that's available, and then that can be used for a small animal trials. Yeah. So the challenge with MPI is that when you scale it up, the scaling up for the system is very, very difficult, because it needs a very strong magnetic field to saturate the particle outside the region of interest.
So it can actually work with small specimen, like I don't know, a mouse, a frog, some small.
At the time, obviously there are groups. They are working on the human scale MPI. I haven't seen their publication, but I know that there are people working on the bigger scale of this MPI, and then upscaling it is a big challenge. So there are people that trying to have the human scale MPI system.
Terrific. And I can actually also ask only one more question. So you said that you actually delivered particle in this deep region on the brain where you can treat Alzheimer. So actually the question now, I will split it in sub questions. So first, how did you cross this brain blood interface? And then second question, what do you actually do to stop Alzheimer progressing? Like, do you kill some, like, pardon my medical ignorance, but what is the enemy here? Are there some cells that you want to kill? Or it's, how does it work? Can you give a bit more information?
Okay, good question. So the first part of the question, if actually I want to tell you, is that we have developed different generation of magnetic actuation schematic. So we are using this on and off system initially, and that helped with moving the particles to some extent. Because if you are using a constant magnetic field, and if the magnetic field is very strong, the particle, they stick to the blood vessels, they don't move. So we started to navigating with, that on and off system. And then we started to use two magnets, so in two different directions. And then we did lots of modeling for that. So that actually magnetic field, which is on and off, is helping the particles to pass through BBB. So that's the reason that we can achieve, the particles are crossing the BBB. In the test that we had, in the in vivo test that we had, there is no control. So you cannot target left or right side of the brain. And the particles are passing through BBB in whole brain area. But they are also reaching to a deeper region, which is of interest to us, which is hippocampus. And then that's the place that the Alzheimer's disease affects. So they are reaching to that area as well. And in Alzheimer's disease, now I'm trying to answer your second question. So in Alzheimer's disease, what we have is a protein. This protein is coming in, obviously my background is also engineering, so I'm actually telling you what the answer that my colleagues from biomedical group, he was giving me. So what the problem is, that protein doesn't let these neuronal cells to communicate with each other. And that is the reason for the Alzheimer. That's one of the prime reasons, because again, that's open. That's an open question in the medical science. So one of the reasons that they think is that specific protein. And the thing is, in vitro studies, in some of those studies that they have done, they find this drug called osmotic. So the drug was actually removing those proteins, was having some sort of effect on that. But the problem is that reaching to that area of the brain and then releasing drug to that specific region of the brain is very difficult. And then with that study, we are trying to, load those osmotic drug to nanoparticles and then using this mechatronic system to reach to the brain and then release it in the hippocampus.
Yeah, great. Awesome. This really sounds like a very interesting research. I haven't done that much medical, but now when I just listen to it, it really feels like you're solving a very, very important problem. And that's all good. Well, thank you, Dr.
Thank you. Thank
Dr.
you
Ali,
for having me. It was very, very nice talking to you.
Yeah, and I hope that we are going to see each other now on the Mars conference. It's been announced today.
Yes, so. it was a very, very exciting and good news for the community of micro-nanorobotics.
True that. Okay, thanks a lot, and hope that we're going to have a talk again at some point in the future when you're going to present some awesome new results.
Thank you. Cheers.